Today we will be discussing the molecular basis of skeletal muscle contraction using the article Regulation of Contraction in Striated Muscle by Gordon, Hampshire, and Regenar, along with Sherwood for background information for foundational understanding prior to breaking down the molecular mechanics. During our presentation, we will be discussing the following objectives: the thin filament regulatory system, actin-activated ATPase behavior, calcium-dependent activation, biochemical reactions, and structural features. In order to understand the molecular mechanisms described within our lecture, we need to have a solid foundation. Skeletal muscle is organized into repeating sarcomeres, which are the functional units responsible for contraction. Each sarcomere contains thick filaments, which are composed of myosin proteins, and thin filaments composed of actin, troponin, and tropomyosin. It is important to remember troponin and tropomyosin are regulatory proteins. The Z lines shown here within the saccular are the ones that anchor the thin filaments, while the A band represents the region occupied by the thick filaments. During contraction, thin filaments slide towards the center of the sarcomere without changing their actual length. This is a process known as the sliding filament mechanism. Excitation, contraction, coupling refers to the physiological process which an action potential triggers muscle contraction by increasing the calcium levels. When motor neurons are stimulated in the muscle fiber, the calcium is released from the sacroplasmic reticulum into the cytosol. The increase in calcium allows calcium ions to build and bind to troponin C on the thin filament, causing tropomyosin to shift and expose the myosin binding sites on actin. Once exposed, myosin heads can attach, forming cross bridges and initiating tension development. We have diagrams showing the structural organization of the thin filament regulatory complex. The large gray spheres shown here represent the actin monomers forming the backbone of the thin filament. Along the actin filament is the tropomyosin, shown as the intertwined strands which block the myosin bonding sites physically in the calcium absence. The yellow, red, and green components in this figure represent the three subunits of troponin complex. The TNC is shown here in red, the TNL in green, and the TNT in yellow. TNC calcium binding site subunits are the ones that initiate activation when calcium ions bind. TNL, shown here in green, is the inhibitory subunit, which is the interacting with actin and tropomyosin in this figure to maintain the blocked state when calcium ion is absent. TNT, here shown in yellow, anchors the troponin complex to the tropomyosin, which helps stabilizes its position along the actin filament. The head-to-tail overlap of the adjacent tropomyosin molecule allows a structural continuity along the filament. In a low calcium state, troponin holds tropomyosin in the blocked position, preventing strong myosin binding. When calcium ions bind to the TNC up here, tropomyosin then shifts into the open state, which exposes the actin site required for cross-bridge formation. In the helix map shown here, this is a deep dive into the alpha helix segments of tropomyosin. This is a long two-chain coil protein. The helices focus and fold into repeating structure molecules along the tropomyosin. Each region colored responds to a specific domain responsible for an action. You have A, B, and C helices interacting closest with the actin, helping the tropomyosin position to the myosin binding sites. D and E helices stabilize the coiled core, maintaining the rigidity. F, G, and H helices interact with the troponin complex, especially troponin T, which anchors troponin to myosin. Understanding how myosin S1 ATPase activity responds to changes in the S1 concentration of regulatory protein conditions are crucial. Panel A shows here plots ATPase activity in animals on the y-axis against the myosin S1 concentration on the x-axis. The positive actin curve shows the ATPase activity increases slowly when actin alone is present. Shown here on the positive ATM curve representing actin plus troposin shows a much higher activation level, describing that tropomyosin enhances the actin ability to activate myosin. The ATM-TN with the calcium ions curve rises the steepest, showing maximized ATPase rates under the full activation with troponin and tropomyosin present and calcium binded. On the other hand, ATM-TN with no calcium condition has a lower ATPase activity because troponin 1 holds the tropomyosin over the myosin binding sites, preventing the cross-bridge cycling. Panel B shows activity at the micromolar ATP per second. This is plotted against the S1 concentration. The points are almost on an exact straight line, showing the ATPase activation is almost directly proportional to the number of available myosin heads. The dashed line, shown here, is the predicted response of each S1 head contributing independently. The closeness indicates that the myosin head operates independently under these experimental conditions. Gordon performed supporting the linear kinetic model of the ATP turnover. Taking a look at the calcium dependence of force and the rate of tension development, panel A in the top left shows the tension time at the PCA 4.0, 6.4, and 6.6. Higher calcium results in faster and larger tension development, demonstrating calcium accelerates the transition to force-generating states. Panel B, shown here, plots normalized KTR versus normalized tension, which is on the x-axis. The steep nonlinear rise reflects cooperative activation. Once part of the filament activates, neighboring regulatory units become easier to activate. Panel C, shown here on the bottom left, is the related KTR on the y-axis to the unloaded shortening of the velocity on the x. The linear correlation suggests the tension redevelopment rate and the filament sliding are common, underlining cross-bridge kinetics. Shown here is a detailed structural mapping of myosin S1 and its actin binding loops. Panel A and Panel B, Panel A and Panel B, showed ribbon models of myosin with specific labeled interaction sites. So here on Panel B, you will have sites as 24 to 25, you have 40 to 45, 90 to 90, 100, 404 to 415, 467 to 678, and 625 to 647. Panel C, D, and E have surface rendering of myosin docked into actin. The colored regions, purple, blue, green, and red, are responsible for both the weak and the strong binding states. These are the contact points. These structural details explain how cross bridges physically attach during the ATPase cycle and why the binding strength changes directly with the nucleotide state. There is a relationship between the unloaded shortening on the x-axis and the shortening duration with length of change, which is shown here on the y-axis, under varying calcium levels, and this is shown right here on this graph. The length of change in micrometers is plotted against the shortening duration in milliseconds. In sex traces of the PCA, 4.5 and 6.0, along with other values that show the higher calcium, the faster and the greater of the shortening. The curvature of each line reflects how fast the shortening occurs. So steeper the line indicates a higher shortening velocity. Shallower the line means a slower shortening velocity. The upward curve demonstrates that the shortening velocity increases with thin filament activation, linking the calcium binding to the mechanical output. With looking at the PCA values or calcium traces by the graph by itself, it shows the mechanical concept that unloaded shortening follows almost a linear relationship during that early phase, but then it begins to plateau at the max of the shortening limit as it approaches. So you have this linear phase and then you have the plateau right here during that singular change. Focusing on the graphed figure here, the left side shows 4 thin filament regulatory states. You have calcium free with no force, calcium bound with no force, calcium bound with force, and calcium free with force. The transition arrows marked here show the rate of constants with K on, K off, G gap, F app, all describing how calcium binding and cross-bridge turnover interconnect these states. Panel A graph figure and panel B graph figure of the right show the non-linear increases in normalized KTR as tension increases. This confirms that the cross-bridge binding enhances thin filament activation. The slopes of these lines on the graph rise steeply as tension increases, showing that the KTR accelerates non-linear with the greater thin filament activation. The upward curvature shows once the cross bridge is bind, it is easier for other cross bridges to then activate. Table 2 shown here on the right-hand side lists the physiological and the biochemical factors increasing the calcium sensitivity of activation. So the factors that enhance calcium binding include increased pH, decreased magnesium, increased temperature, which all make troponin C bind calcium more easily. The factors on table 2 which promote myosin binding are increased sarcomere length, decreased lattice spacing, activation or neighboring cross bridges, and MRLC phosphorylation, which make actin more available for that myosin attachment. On the left of this diagram, it shows the complete ATP-ACE reaction pathway. It begins with the AM and ATP, causing an actomyosin dissociation, followed by ATP hydrolysis, weakening, binding to actin, phosphate release, then the power stroke, and then the ADP release. This scheme here shows the biochemical sequence which underlies each cross-bridge cycle. Here on table 1, this shows the rate of constants for each ATPase reaction step under a different temperature, iconic strengths, and preparations including the actomyosin S1, HMM, myofibrils, and fibers. ATP cleavage, phosphate release, ADP release, and steady ATPase are the key rates and reveal how environmental changes alter the kinetic cycling. These two figures combined model the understanding for force generation at the molecular level. Here we have a simple comparison of the thin filament regulatory structure in calcium-free and calcium-bound state. We have calcium-free here on the left. So here you have the TNL holding the tropomyosin, which is shown here as TM in the blocking position. T1 and T2 regions of troponin T, the labeled cis-190 marker, which is used to orientate the model, and the head to tail overlap of tropomyosin is going to stabilize that off state. On the right side, you have this where calcium is binding to the TNC, and the troprinone shifts, reducing the TNL's inhibition and allowing the tropomyosin to then move away from the myosin binding sites. The same structural labels, the T1, T2, cis-190, and overlap, are all still present, but the support on the state where myosin is able to generate force.